Good morning, grade 8 students. In this presentation, we're going to talk about the characteristics of the specific immune response. So let's start with the characteristic of the specific immune response. But before we start, we have to talk about the lines of defense. First line of defense is the natural barriers, which represent the first line of defense. If this line was crossed, we move to the second line of defense, which is the natural barriers. And if this the second line of defense, the non-specific immune response, is crossed, we move to the third line of defense, which is represented by the specific, either cell-mediated or the humoral. Cell-mediated immune response includes the T lymphocytes, where the T lymphocytes, they are the main actor cells, they kill other cells. While this humoral immune response, we have the B lymphocyte, they are the active cells. So let's talk about the characteristics. We have to differentiate between the primary and the secondary immune response regarding latency period, amount of antibodies, and the duration of the response. Let's keep in mind first, what is the primary immune response? Primary immune response is the immune response upon the first contact with the pathogen. So the first time I saw the pathogen, it's called the primary immune response. Whereas the secondary immune response is the immune response upon the second contact, but with the same pathogen. For example, I saw the flu virus for the first time this year. So this is called the immune response against it. It's called the primary immune response. When I saw it again for the second time, this immune response is called the secondary immune response. Since I saw the same pathogen for the second time, it's called the secondary immune response. So what's the difference between the primary immune response and the secondary immune response in their characteristics? First, we have recognition. So we recognize the antigen. For example, we recognize the flu virus antigen. And then the cells, they proliferate, they increase in number. This is called multiplication, or it has another name, it's called proliferation or amplification. These three terms, proliferation, amplification, multiplication, all of that, we have the same meaning, meaning that increase in number. And then we have differentiation into either effector cells or memory cells. And we will see what is the role of the memory cells. As I said, multiplication, it has the same meaning as proliferation. So multiplication, proliferation, amplification. These three words, they have the same meaning, meaning that increase in number, and the cells, they are dividing and increasing in number. Antibody secreting cells. Antibody secreting cells, they are called now plasma cells. We don't call them B lymphocytes. So plasma cell is the mature one of the B lymphocytes. So during differentiation, as we saw, the cell either differentiate either effector cell or memory cell. If the effector is B cell, so we call it a plasma cell, since it's able to secrete antibodies. While the effector T cells, they are called killer cells. Immediately will kill other cells. So, as I said, multiplication means proliferation, means amplification. These three words, they have the same meaning. Antibody secreting cell is called the plasma cell. Effector B cells, because they secrete the antibodies, we call them the plasma cells, while the effector T cells, they are called the killer cells. This graph is showing the difference between the primary immune response and the secondary immune response. First time we inject the antigen, this graph is showing the rate of antibodies in the blood in arbitrary unit, AU, as a function of time in days. So first, the first injection of the antigen, we call it the primary immune response against it. While after the second injection, we call it the secondary immune response. If you look at the graph here, the primary immune response, it starts after 10 days of the injection. Injection at day 0, the immune response starts at day 10. So the latency here, the latency is the time needed to start the immune response. So the time needed is 10 days. So we call the latency is 10 days in the primary. While in the secondary, the second antigen was injected at day 50. So it needs about 2 to 3 days to start the immune response. So the latency here is 2 to 3 days. While the amount of antibodies in the primary immune response, it was 2 AU, while in the secondary immune response, it was about 6 AU. So in the secondary, we have higher amount of antibodies. The primary immune response, it starts at day 10 and ends at day 40. So the time or the duration, it's from day 10 to day 40, it's 40 minus 10. So the duration will be about 30 days. Why? The secondary 52 to 90, so it's about 38 days. So it's longer time of protection. So the latency is about 10 days in the primary immune response. It needs 10 days to start. So it's a long latency. Why? In the secondary, it's a short latency. It needs only 2 days. In the primary immune response, we have low amount of antibodies to AU. While in the secondary, we have high amount of antibodies. They are about 6 AU. The primary short duration of protection or short duration of the response starts at day 10 to day 40, so about 30 days. While the secondary, we have long duration of response, and here it continues afterwards since the amount of antibodies they didn't drop to zero. So that is the difference between the primary and the secondary. Primary and secondary. This table is a comparative table of primary and the secondary immune response. Primary latency is the time needed. You have to know this. You have to know the definition of the latency. Latency is the time needed to start the immune response. Amount of antibodies here, duration of the response. The primary, the latency is long. It needs about 10 to 12 days. So here, it's 12 days according to the example. So you have to say it's long latency. While at the secondary, it's a short latency. Amount of antibodies in the primary, we have low amount of antibodies, whereas in the secondary, we have high amount of antibodies. Duration of the response, it's short duration during the primary, while during the secondary is a long duration of the response. So here, let's do a quick revision of what we have explained before. So you have to know first, what is the primary immune response? We said that the primary immune response is the immune response upon the first contact. So I saw it before the first time, this pathogen that can cause a disease, it enters my body for the first time, the immune response against it is called primary immune response. While the secondary immune response is the immune response upon the second contact with the same antigen or the same pathogen. We said that first we recognize the antigen, then we have multiplication or proliferation, then differentiation to either effector cells or memory cells. So we said that multiplication or proliferation or amplification, all of them, they have the same meaning. Antibody secreting cells, we call them as plasma cells. Effector B cells, they are the plasma cells, and the effector T cells, they are called the killer cells. We said the primary immune response and the secondary immune response, differences between them, long latency in the primary, short latency in the secondary, high amount of antibodies in the secondary, low amount of antibodies in the primary. We said short duration of protection in the primary immune response, while long duration of protection in the secondary immune response. So, what is the role of the memory cells? What do they do inside the body? Primary immune response takes a long time to eliminate the infection, yet it produces specific B and T cells. These are called memory cells. These memory cells, they memorize, they knew that this antigen is causing harm for our body, is causing diseases, and this one should be eliminated immediately. So the primary immune response will take a long time, but it produces memory B and T cells. These memory B and T cells, they recognize the antigen and they are needed for the next invasion. When this virus or this bacteria comes again, these memory cells, they knew it, they memorize it, they knew that this one is causing disease. So you have memory cells and you have B and T effector cells. So you have more number of immune cells so that the immune response in the secondary will be more efficient, will kill the pathogen and eliminate it in a shorter period of time. Why? Because we have the memory cells. So primary immune response takes a longer duration, yes, but it produces the memory B and T cells. These are important for the secondary immune response to eliminate the pathogen immediately and after a shorter period of time. So this is the importance of the memory B and T cells. So what are the results of the primary immune response? Results, we eliminate the foreign antigen, even though after a long period of time, but we eliminate it, we get rid of this foreign antigen, and we produce memory B or T cells. What is the benefits of, what are the benefits of memory B and T cells? They are used in the secondary immune response, where they recognize the antigen immediately and eliminate it in a shorter period of time. What are the advantages of the secondary response? It's faster, it starts faster, less delay, amount of antibodies, they are higher in the secondary, and duration, it stays for a long period of time and protects my body for a long period of time. So this is the importance of the memory B and T cells. So memory B and T cells, they are needed, they are produced in the primary and they are needed in the secondary immune response to kill the foreign antigen immediately and in a shorter period of time. This is the benefits this is the benefit of the memory B and T cells. So here the memory B and T cells, they are needed in the secondary, but they are produced in the primary immune response. So memory B cells and T cells, they are needed in order to help to recognize and to get rid of the foreign antigen immediately after a short period of time. Okay? Thank you.